before, scientists working in one of the harshest environments on Earth uncovered a fossil so unexpected, so fragile, and so scientifically disruptive that it took over more than a decade just to understand what we were looking at. This creature walked upright, not like us. She climbed trees, but not like apes. She lived in forests, not grasslands. And in doing so, she shattered some of the most deeply held assumptions about how, where, and why humans evolved. Is Ardipithecus ramidus, known to the world as ARDI, a species that lived 4.4 million years ago, just after the split between the human and chimpanzee lineage. And ARDI doesn't just tell us where she came from, she tells us what we're not. Let's dig into it. ARDI was discovered in Ethiopia's Afar Depression, a geologically active region where the tectonic forces pull Africa apart, exposing deep layers of ancient sediment. This region has produced some of the most important fossils ever, such as Lucy, Salam, Australopithecus anamensis, and even early Homo. But ARDI was different. The remains were scattered across the landscape of soft, crushed sediment. The bones were so fragile they would crumble if touched. Researchers couldn't simply dig her out. They removed entire blocks of earth, encased them in plaster, and transported them intact, a process that took years. This excavation took 15 years. But why? It wasn't just an excavation. Each bone had to be stabilized chemically, scanned with high resolution CT imaging, reconstructed, and then physically reassembled. Many bones were crushed almost flat by geological pressure. Others were warped, fragmented, or distorted beyond recognition. This wasn't just a simple fossil paper. This was an entire science special issue in 2009. More than a dozen papers, each addressing a different part of ARDI's anatomy and environment. Before ARDI, most models of human evolution assume something very specific, that our ancestors evolved from chimp-like creatures that lived in open savannas, walked on their knuckles, and gradually stood upright to see over the tall grass. ARDI demolished that idea. She wasn't knuckle walking, she wasn't adapted to savannas, and she wasn't chimp-like in the way that we once assumed our ancestors must have been. In fact, ARDI suggests something, radical. Chimpanzees may be more specialized than we are. The last common ancestor of humans and chimps probably didn't look much like a modern chimp at all. ARDI's hands tell a subtle but powerful story. She had long, curved fingers, excellent for grasping branches, but her wrist bones lacked the locking mechanism seen in knuckle-walking apes. No reinforced knuckles, no weight-bearing adaptations. This means knuckle walking evolved after the human-chimp split in the chimp lineage. Her arms were long, but not built for brachiation like Gibbon's. She wasn't swinging, she was climbing, cautiously, moving deliberately in a three-dimensional forest environment. The pelvis is where ARDI becomes revolutionary. The upper pelvis, the ilium, is short and broad like ours. This repositions gluteal muscles, allowing them to stabilize the body during upright walking. But the lower pelvis, the ischium, remains long and powerful. That means strong hip extensions for climbing. This is not half-baked bipedalism. This is dual-function anatomy. She can walk upright, but slowly and ineffectively. But she can also climb, but not like a modern ape. foot might be the most visually striking compromise in her body. She had a fully grasping big toe, capable of gripping branches. But unlike chimps, the rest of her foot was stiff. The lateral foot bones show adaptations for pushing off during upright walking. She didn't have arches like us, but she wasn't flat footed like other chimps. She walked upright without sacrificing her grip on the trees. The teeth preserve behavior better than almost any other part of the skeleton, and Artie's teeth are quite revolutionary. Her canines are small, blunt, and show very little difference between male and females. In living apes, large canines are weapons, signals of defense in male competition. Artie didn't have those weapons. What does this mean? It suggests reduced male aggression, possibly more pair bonding, possibly less violent competition for mates, all millions of years before large brains evolved. Her teeth were generalized, not specialized for tough grasses, not optimized for fruit alone. She likely ate fruits, leaves, seeds, and soft plant matter. Granted, these look much different than what we have today. Think about Artie's environment contradicts the savanna hypothesis. Plant fossils show figs, palms, and hardwood trees. Animal fossils include columbine monkeys, forest birds, and woodland antelope. Carbon isotope analysis confirms a C3 dominated ecosystem, forest plants. She walked upright in the shade. Where does Artie belong? She sits near the very base of the hominin lineage. There are three interpretations: direct ancestor, Ardipithecus, Australopithecus, Homo, a very close cousin. Not ancestral, but nearly identical to the ancestor. Or an early experiment, a branch that didn't survive. Most researchers tend to favor the first two. And either way, Artie changes the baseline. She shows us what hominins actually look like. Ardipithecus ramidus reminds us that evolution doesn't move in straight lines. It tests possibilities, explores compromises, and discards what doesn't work. Artie wasn't becoming human, she was becoming Artie. And from forms like hers, our lineage eventually emerged. In the next chapter of this story, we meet Australopithecus anamensis, a species that takes upright walking even further. Subscribe for more deep dives into human origins here on Fossil Matrix.